Hi, today we're going to be studying evolution, the history of life on Earth as a review with chapter 17. Most of what we do today should be a review, so make sure that you take good notes. Tomorrow in class you'll have a little mini quiz just seeing how you do on this. Okay, so get ready to take notes, chapter 17. We're going to start with the origin of the universe, which was started by the Big Bang. And so all of the research that has been done is showing that the universe is expanding. Instead of getting smaller, it's getting bigger and bigger. And they've been able to test that with different evidence that is coming in from outer space. Using the Hubble telescope has been very helpful with that. So 15 billion years ago is the best estimate as to when the universe began. And then after that, it was 4.6 billion years ago is when the Earth formed. So remember, we're saying between 4.5, 4.6 billion years ago is when the Earth formed. And so let's talk briefly just about how those ages are determined. So what they do when they determine these ages is use what's called radioactive dating. You might want to go ahead and jot that down. Radioactive dating. So they can determine how old a rock is or a fossil is by how much radioactive material it has in it. There's different elements that they use to do that, but basically what you need to know is they're looking at a ratio of radioactive elements present before and after. So they're looking at that ratio, and that ratio tells how old the Earth is, how old that Earth is, or how old that rock is, or how old that fossil is, whatever you're trying to determine the age of. Okay, so if we look at the early Earth, like you watched in the video, you know that from the video that it was very, very hot. And a lot of that heat energy came from that radioactive decay. Remember, that radioactive decay is what we just talked about with determining the age. They're using that ratio of how much radioactive decay it has in it to determine how old the Earth is. But it also releases a lot of heat. The atmosphere itself was very different. It was made up of many different elements. So the early atmosphere would have been made up of elements like nitrogen, compounds like carbon dioxide, water, hydrogen, carbon monoxide. There was no ozone layer. And then some research says that there might have been ammonia and methane in the atmosphere as well. So things like nitrogen, carbon dioxide, water, hydrogen, carbon monoxide, ammonia, and methane, with no ozone layer being present. There was also no free oxygen available, so there was no oxygen for like we need to breathe. They did not have that available. It was only in compounds. So now, the first life came about 3.5 billion years ago as the oldest fossil that they have tested for that early bacteria. First life. All right, so let's talk about the beginnings of life. We know that life on Earth was very hostile. There were so many volcanic eruptions going on at the time. It was very hot. It wasn't protected very well. So by that ozone layer that we talked about that protects us from radiation. So how did the life originate? We talked the other day in class about the difference between a hypothesis and a theory. So remember that these are only hypotheses, which are testable explanations. So these are three hypotheses that are being tested. One is that life originated from another planet. It came to us based on from meteors or asteroids that hit the Earth and has amino acids and other compounds in it. One is unknown. Obviously that's not testable, but that still means that there's still research being done to see what else it could be. And then third is that there's a non-living substance that interacted with the environment, and that's what we're going to spend most of our time on. So Operin and Haldane are two scientists that were working independently but came up with a similar hypothesis for how life on Earth would have begun. And that hypothesis is called the heterotroph hypothesis. There's three parts to the heterotroph hypothesis that we'll talk about. The first part is that there has to be a supply of organic molecules. Organic molecules means that they contain carbon. The second one means that there's processes that have to assemble those molecules into polymers. So polymers are really big molecules. So you have to have a process that takes those small molecules from number one and makes them into really big molecules. And then the third thing is that those polymers have to be able to reproduce in some way. So let's talk about each of those in a little bit more detail. So the organic molecules come from a experiment done by Miller and Urey that they talked about on the video. And that recreated conditions that resembled the early Earth. So they took this hot heatness, because remember how we talked about how it was much hotter on Earth then than it is now. So they took some ocean and they boiled it to make it hot. And then they removed they removed the atmosphere that we have, and they put in the atmosphere that they believed that they had. So we talked about how that was nitrogen, carbon dioxide, water. They also thought that there was a lot of ammonia and methane. And then they provided this spark, and the spark was supposed to represent lightning. Remember we talked about how there were all those storms that were going on? They then cooled it down because the earth cooled, and then they had this collecting trap. And in this collecting trap is where they found organic molecules. So they found a lot of those organic molecules in that collecting trap. There's a couple questions that have come up about their experiment since then, and that would be that they think that the atmosphere might be different now than it was then. But it has been retested with new atmospheric conditions and have gotten the same results. Also, they think that there's too much energy supplied, so you might not have gotten it in the quantity that they got it. But you're still able to get the organic molecules. Okay, the second part is the formation of the polymers. And that was done by Carnes and Smith. And so what they studied were clay particles that were on the Earth's surface. Those clay particles had a repeating crystalline structure, and then that crystalline structure allowed those amino acids that we just talked about for Miller and Urey to be attracted to them because they have charges on them. So like a positive charge is attracted to a negative charge. And then those crystals help catalyze. Catalyze means to help. So those crystals catalyze the bonding of small molecules together. So all those small molecules that were attracted to the clay in the first place the clay now helped form bigger molecules. Okay, so then the third part is the replication system. And that's kind of like asking the question, what came first, the chicken or the egg? And so our question is, what came first, DNA or protein? And well, actually the answer is that most scientists now think that RNA came first. And there's a couple reasons for that. One is that RNA can replicate itself, and two is that it can catalyze reactions. Remember, catalyze means to help out with reactions. So it's when we're forming proteins, DNA gets converted into RNA, and then RNA gets converted into the protein. So RNA is like the intermediary in between those two. So most scientists now agree that RNA was probably the first molecule. All right, the next thing that we need to talk about then is biological evolution. So we have the first cells, but let's define what life is. So what is life? There are several characteristics that life has to have. One is that the molecules need to be able to reproduce. They have to undergo mutations, so that means that they change. And then there has to be natural selection. Remember, we talked a lot about natural selection with our last unit, so the environment choosing the traits that are most likely to survive, and then remember that those then have to reproduce. So all three of those things are tied in together. When we talk about life is now guided by what's called the cell theory. So in order to be considered a living thing, you have to abide by the cell theory. And there's three parts to that theory. One is that all living things are made of cells. The second is that cells are the basic unit of structure and function. And the third is that cells come from other cells. So those three things, if an organism meets those, or if an item, I guess, meets those characteristics, then they are considered living organisms. All right, so let's talk a little bit about the evidence of these organisms forming, and those are by what are called microfossils. You listened about microfossils on the video, and they were tiny single-cell filaments, and these are the ones that are the oldest ones that have been tested and found to be 3.5-ish billion years old. And they're found in stromatolites, and if you remember from the video, stromatolites were those layers in the rock that contained microfossils, and they were struggling to get to the light so they could go through photosynthesis. And so they left layers where they were found. And so those have been tested to be actually made of cells. All right, the type of cell that they were is what's called a prokaryotic cell. A prokaryotic cell is a cell that does not have a nucleus, does not have any organelles. Like I'm sure you've heard about cells before, so remember some organelles are like mitochondria and ribosomes. If you don't know what those mean yet, don't worry, you will soon. These are very small organisms. The very first ones would be what we call methanogens, which means that they live in an area with a high concentration of methane. The first ones we found were about 3.5 billion years ago, like we talked about before. And then 2.2 billion years ago, we had our first photoautotrophs. Photo means light. Autotroph means that they can make their own food. So these were the first organisms to go through photosynthesis. And they were also bacteria. All prokaryotes are bacteria. So remember when we talked about how photosynthesis gave us to have our first free oxygen available? That free oxygen helped spur a lot of life, like we talked about, how life pretty much was able to spread across the whole planet. But it also caused a mass extinction, the first mass extinction on the planet, because that free oxygen was toxic to a lot of the organisms, so they weren't able to survive in that environment. All right, so then after the prokaryotes, then came the eukaryotes. And the eukaryotes are what kind of cells that you have. So that was about 2 billion years ago. And these cells have a nucleus, they have organelles, and they're much larger than prokaryotes. So kind of the opposite of the characteristics that we just talked about. And the way that they formed is called the endosymbiont hypothesis, put out by Margulies. The endosymbiont hypothesis. So the first thing you need to know is what symbiosis is. Symbiosis means that it's two organisms that are living together. Two organisms living together. So endosymbiosis means, endo means inside. So endosymbiosis means that you have a cell that lives inside of another cell. So the definition of endosymbiosis is eukaryotic cells evolved from prokaryotes living symbiotically inside of other prokaryotes. So that means that a bigger prokaryote swallowed a smaller one, but it didn't get digested, so it was able to survive. So let's talk about the evidence. You need to write this evidence down. The first thing is mitochondria and chloroplast. Okay, so mitochondria and chloroplast provide our evidence for this, and that's because they are thought to have been prokaryotic cells billions of years ago. So the evidence that mitochondria and chloroplast are prokaryotic cells is that they have their own DNA. And they have their own DNA that looks like prokaryotic DNA, because prokaryotic DNA and eukaryotic DNA is very different. We'll talk about the differences later, but for now you just need to know that it's different. And it's prokaryotic DNA. They can also replicate themselves, so signifying that they were at one time a cell, that they can replicate themselves because they have that characteristic of life that we talked about earlier. Another example or another piece of evidence is that they are both about the same size as a prokaryotic cell. Another piece of evidence is that they have their own ribosomes, which means that they can make their own proteins. And they both have a double membrane, just like a cell has. So that's the evidence for endosymbiosis. Make sure that you have all of that evidence recorded in your notes. What I would like you to do now in your notes is to write down 2 to 3 questions that you have about the notes or about this unit in general that we have completed. So 2 to 3 questions that we will talk about tomorrow in class. Make sure that you have those recorded in your notes as well, and that you bring your notes to class tomorrow. Thanks. Have a great day.